Hey, welcome to Following Science, the podcast where we delve deep into the fascinating world of scientific discovery and innovation. I'm your host, Ritu Parl, and today we're going to be exploring the most fundamental areas of biology, starting with cytology, the study of cells. Whether you're a student, a researcher, or simply a science enthusiast, this episode will provide you with a comprehensive understanding of cytology, covering its history, techniques, applications, and recent advances. So let's dive right in. Cytology is a vital field in biology because cells are the basic building blocks of all living organisms. Understanding cells and their functions is crucial for many aspects of science and medicine, from diagnosing diseases to developing new treatments and therapies. In this episode, we're going to explore the rich history of cytology, discuss the detailed structure and function of cells, and, of course, delve into the various techniques used by cytologists, and lastly, look at the wide range of applications for this fascinating field. So, what exactly is cytology? At its core, cytology is the study of cells. This includes their structure, function, and chemistry. Cells are the smallest units of life that can function independently, and they come in a wide variety of shapes and sizes, each tailored to their specific role within an organism. Cytology encompasses everything from examining the physical characteristics of cells under a microscope to analyzing the complex biochemical processes that occur within them. It's also important to differentiate cytology from histology. While cytology focuses on the study of individual cells, histology deals with the study of tissues, which are groups of cells that work together to perform specific functions. Now, of course, both fields are closely related and often overlap, but they each provide unique insights into the biological world. Now, we can cover histology next time, but let's keep diving into cytology. Starting with the history, which dates back to the 17th century when scientists like Robert Hooke and Antoine Leeuw van Hooke made groundbreaking observations using early microscopes. Now, Hooke is credited with coining the term cell after observing the cell walls of cork tissue, while Van Leeuwenhoek was the first to observe living cells, including bacteria and sperm cells. These early discoveries laid the foundation for modern cytology and opened up a whole new world of microscopic life. Now, cytology has several branches. Each focuses on different aspects of cell study. Descriptive cytology, examines the structure of cells, often using various staining techniques to highlight specific components. Now, functional cytology studies the biochemical processes and functions of cells, such as energy production and protein synthesis. And lastly, diagnostic cytology applies these principles to medicine, using cell analysis to diagnose diseases like cancer. Now that we have a basic understanding of what cytology is, let's delve deeper into the structure and functions of cells. There are two main types of cells, prokaryotic and eukaryotic, which I'm sure you all already know. Now, prokaryotic cells, just to give you guys a brief summary, such as bacteria, are just simpler and lack a defined nucleus. Their genetic material is free-floating within the cell. Eukaryotic cells, on the other hand, which makes up plants, animals, fungi, and protists, are more complex and have a defined nucleus that houses their genetic material. Now, let's start with the nucleus, often referred to as the control center of the cell. The nucleus contains the cell's DNA, which carries the genetic information necessary for the growth, development, and reproduction of the organism. The DNA is also stored within the nucleus. Next, we have the mitochondria, which I am sure that you all know as the powerhouse of the cell, which, of course, it really is. But mitochondria is also so much more than that. It's responsible for producing ATP, which is also where it gets its name, powerhouse of the cell, which is the cell's main energy currency. And it does this through a process called cellular respiration, which is literally the opposite of photosynthesis. So if you know photosynthesis, literally reverse the process and you have cellular respiration. Now, mitochondria also has their own DNA, which is super cool, which is also like chloroplasts because they have their own DNA and it has something to do with the endoplasmic theory. But basically, because they have their own DNA, it's inherited maternally and it's separate from the DNA found in the nucleus, which is super cool because then it's kind of like, oh, this disease is not in the DNA in the nucleus, it's actually in the mitochondria and it was inherited maternally. Obviously, it can be inherited by the father figure, but that happens super, super rarely, and it's normally just the maternal figure. Then there's ribosomes, which is another essential component of the cell. These small structures are the sites of protein synthesis, where amino acids are assembled into proteins based on the instructions encoded in the cell's DNA. Ribosomes can be found floating freely in the cytoplasm or attached to the endoplasmic reticulum, forming what is known as the rough ER. So there's two types of ribosomes, and I'm not going to go into much detail right now, but there's the smooth ER and the rough ER. Now, the endoplasmic reticulum itself, or the ER, comes in two forms, as I just mentioned before, rough and smooth, and they have ribosomes attached to it, which is kind of what makes a rough ER the rough ER, because it just looks like it has dots outside of it, and it's not really dots, it's ribosomes, but yeah, that's what it is. And of course, rough ER and smooth ER have different functions, and for example, rough ER is involved in the synthesis and processing of proteins. The smooth ER, on the other hand, lacks ribosomes, which is why it's just responsible for the synthesis of lipids, metabolism of carbohydrates, and detoxification of drugs and poisons. Now, I said just involved in this, but honestly, the smooth ER is probably involved in tens of thousands of things that rough ER is not involved in. And now, I know you guys know that tens of thousands is an over-exaggeration, but I just want to make it clear. Now, let's move on to the Golgi apparatus, which I like to call Go Food, just to remember what it is like, because it's kind of like Go Food in the sense it transforms the food to another place, which is why it's like going. I don't know if that makes sense. But basically, Golgi apparatus functions as the cell's post office, modifying, sorting, and packaging proteins and lipids for delivery to their final destinations. Proteins and lipids produced in the ER are then transported to the Golgi apparatus in vehicles, or that's what I call it, but in reality it is called vesicles. And in this process, they undergo further processing before being sent to their target locations within or outside the cell, which is why vesicles are so important, because they can literally transport you anywhere in the cell. Well, not you specifically, but you get what I mean. And these vesicles can just bind with the cell membrane to push you out, or they can just bind to anything else to make sure you're being transported where you need to be, like the cells. Now, let's move on to lysosomes, which is the cell's recycling centers. Lysosomes contain enzymes that break down waste materials and cellular debris, whereas different recycling centers, such as peroxisomes, contain enzymes that detoxify harmful substances and metabolize fatty acids. These organelles play a crucial role in maintaining cellular health and preventing the accumulation of toxic substances. And now let's move on to the cytoskeleton. I know it's a lot, but hang with me. This is the last three. But the cytoskeleton is basically a network of protein fibers that produces and provides structural support. Actually, just provides structural support to the cell and facilitates movement. It consists of three main components, such as microfilaments, intermediate filaments, and microtubules. Microfilaments are involved in cell movement and shape changes. Intermediate filaments produce and provide, or rather just provide, mechanical strength, and microtubules serve as tracks for the movement of organelles and vesicles within the cell. The cell membrane, also known as the plasma membrane, is a selectively permeable barrier that surrounds the cell. It is composed of a phospholipid bilayer, which embedded with proteins is basically going to regulate the movement of substances in and out of the cell. And I know you guys know about the cell membrane, so I'm not going to go into much detail, but it also has a crucial role in cell communication and signaling. And lastly, let's go into cell division. Again, I am not going to go into much detail. If you want, I can write another episode about this. But right now, I'm going to focus on cytology, so let's wrap things up. But basically, cell division is a fundamental process for growth, development, and reproduction. In eukaryotic cells, this process occurs through mitosis and meiosis. Mitosis results in the production of two genetically identical daughter cells and is essential for growth and tissue repair. Meiosis, on the other hand, produces four genetically diverse gametes, sperm or eggs, and is crucial for sexual reproduction. Now let's move on to cytology, because I know that's what we've been waiting for. Cytologists use a variety of techniques to study cells and their functions, so let's dive into that. One of the most fundamental tools is microscopy. Light microscopy uses visible light to illuminate and magnify specimens, allowing scientists to observe cell structures in detail. Electron microscopy, which includes transmission electron microscopy or TEM and scanning electron microscopy or SEM, uses beams of electrons to achieve much higher magnifications and resolutions, revealing ultra-structural details of cells. Now, staining techniques are also essential in cytology, as they enhance the contrast of cell components under the microscope. Hemaoxytolin and eosin, or H&E, which is so much easier to say, staining is one of the most common methods, highlighting nuclei in blue and cytoplasm in pink. Special stains such as gram staining are also used to differentiate bacterial cell walls and other specific cellular components. Now, cell culture is another critical technique, allowing scientists to grow and study cells in vitro, or outside their natural environment. This technique is widely used in research and medicine, enabling the study of cellular processes, drug testing, and the production of biological products like vaccines. Flow cytometry is also a powerful technique for counting, sorting, and analyzing cells in fluid suspension. It uses laser-based technology to detect and measure physical and chemical characteristics of cells, such as size, complexity, and the presence of specific markers. This technique is widely used in research and clinical diagnostics. Now, cytogenetics is the study of chromosomes and their role in heredity and disease. Techniques like karyotyping, which involves arranging and analyzing the complete sets of chromosomes from cells, and fluorescence in cytohybridization, which is basically FISH, which uses fluorescent probes to detect specific DNA sequences. And they're all used to diagnose genetic disorders and study chromosomal abnormalities. Now, cytology has a wide range of applications, particularly in the field of medical diagnostics. One of the most well-known applications is the Pap smear, a screening test for cervical cancer. In this test, cells are collected from the cervix and examined under a microscope for abnormalities that may indicate precancerous or cancerous conditions. Now, they also use cytology in fine needle aspiration, which is basically another diagnostic technique, and is used to obtain cells from tumors or other lesions for examination. A thin needle is then inserted into a specific area or just a suspicious area, and cells are then aspirated and analyzed to determine whether the lesion is benign or malignant, which is often used in cancer. Now, in research, cytology is used to study cell physiology and pathology, providing insights into cellular processes and disease mechanisms. It plays a crucial role in cancer research, helping scientists understand how cancer cells differ from normal cells and how they respond to treatments. Cytology also contributes to drug development by examining and enabling the screening of potential therapeutic compounds. Now, cytology is also used in criminal investigations, and if you're wondering how, well, it's through forensic cytology, and this is where cell analysis can provide crucial evidence. Because techniques like DNA fingerprinting rely on the analysis of cellular material to identify individuals and establish links between suspects and crime scenes. So basically, if you want to go into forensics, this episode is good for you too. And then there's environmental cytology, which basically studies the impact of environmental factors on cell health. For example, researchers may examine the effects of pollutants on cell structure and function, providing valuable information for environmental protection and public health. Now, cytology also has applications in agriculture, where it is used to study plant cells and improve crop yields and resistance to diseases. Techniques like protoplast fusion, which involves the fusion of plant cells without cell walls, are used to create hybrid plants with desirable traits. Now, the field of cytology is continually evolving, with new technologies and techniques pushing the boundaries of what we know about cells. One of the most exciting recent advances is in the development of single-cell RNA sequencing. This technique allows scientists to analyze the gene expression profiles of individual cells, providing unprecedented insights into the cellular diversity and function. Now, single-cell RNA sequencing has revolutionized our understanding of complex tissues and has important implications for cancer. And, of course, developmental biology. Now, another significant advancement is the use of CRISPR-Cas9 technology for genome editing. Now, I've already mentioned this a lot in my previous episodes, and I also have just another episode on gene editing that I did some while ago. So if you want to learn more about this, make sure to check that out. But I'm just going to move on. But then there's high-throughput imaging techniques, which have also transformed cytology. Automated microscopes and advanced image analysis software can now capture and analyze thousands of cells in a short period, providing detailed insights into cell populations and their behaviors. These techniques are particularly valuable in cancer research, where they can be used to identify rare cancer cell subpopulations that may be resistant to treatment. Now, organoid technology is another exciting treatment and development. Now, you might be wondering what organoids are, because I know I sure was when I first researched it. But organoids are basically miniature three-dimensional structures grown from stem cells that replicate the architecture and function of real organs. Crazy. Now, these models provide a more accurate representation of human biology than traditional cell structures, making them invaluable for studying disease mechanisms and testing new drugs. Now, lastly, because we have to talk about AI wherever we go, the integration of AI and machine learning in cytology is opening up new possibilities. AI algorithms can now analyze complex datasets, such as those generated by single-cell RNA sequencing or high-throughput imaging. Now, to identify patterns and make predictions about cell behavior, this can all be used, which is crazy. Now, these technologies are also helping to accelerate discoveries and improve the accuracy of diagnostic techniques. Advances in nanotechnology are also improving and impacting cytology. Nanoparticles can be designed to target specific cell types, delivering drugs or imaging agents directly to diseased cells while sparing healthy ones. This targeted approach holds great promise for improving the efficacy and reducing the side effects of treatments for diseases like cancer. Now, lastly, and I know I promised that the last one would be like 10 minutes ago, but lastly, and I swear this is the last one, the development of lab-on-a-chip technologies is making it possible to conduct sophisticated cell analysis on tiny portable devices. This microfluid system can manipulate and analyze single cells or small cell populations with high precision, enabling rapid and cost-effective diagnostics and research. Now, lastly, let's look into the future. And when I said lastly, I did mean lastly for that specific section, and this is our last section now, so let's go into it. Now, as we look to the future, the field of cytology is poised to continue its rapid advancement. One of the most promising areas is personalized medicine, where treatments are tailored to the individual patient's cellular and genetic makeup, making the entire treatment just more effective for them particularly, which is great because then that ensures that that person's recovery will be the best it can be because of that personalized treatment. Another exciting direction is the study of human microbiome, which is the community of microorganisms that live in, on our bodies, which is crazy to think about. Like, imagine just looking at your skin under a microscope and you see all these like microbiomes just formulating on your skin. Now that I think about it, it does sound kind of gross, but it's also so cool because understanding how these microbial cells interact with our human cells is crucial for developing new therapies for a wide range of conditions, from digestive disorders to mental health issues, which basically means that the things you could find gross are actually helping you. I know, it's amazing. Now, stem cell research is also expected to yield significant breakouts, or rather breakthroughs, and I mentioned this a lot before, talking about how just cancer cells can be more like predicted and just helped because of cytology. And this is exactly what I mean by stem cell research expecting significant breakthroughs. Because by understanding how stem cells differentiate into various cell types, scientists hope to develop regenerative therapies that can help replace damaged tissues and organs. This could have transformative implications for treating conditions like spinal cord injuries, heart disease, and diabetes. Now, there's a lot more we can talk about cytology, but I don't want to keep y'all here for too long because I know at some point it can feel like I'm droning on and on, so we'll cover the rest in another episode. Which is why that brings us to the end of today's deep dive into cytology. We've covered a lot of ground, from the basic structure and function of cells to the advanced techniques used to study them, and literally everything else we could cover in this episode. Now, I want to thank you all for joining me on this journey through the microscopic world. If you have any questions or comments, feel free to reach out in the comments. Now, of course, don't forget to subscribe to Falling Science for more episodes on fascinating scientific topics. Until next time, keep exploring and stay curious. This is your host, Curious George. No, just kidding. Virtual Parle, signing off. Have a great day.